Good afternoon, everybody. My name is Cody Green, and I am working in conjunction with Dr. Jamie Wallen and Dr. Maria Gainey to bring you a presentation on Repressor Bioinformatics. So a little bit of background information, starting with bacteriophages. Bacteriophages are viruses that infect bacteria, certain bacteria, as you'll see here shortly. They store their DNA or RNA, depending on what type of virus they are, in the capsid head. The capsid head is this piece right here. It's the protective shell that encodes for, or that protects this DNA right here. My eraser. So bacteriophages use their tail fibers and spikes. See here, this is a tail fiber, and this is a spike, to latch onto the outside of the bacterial membrane, and they will then inject their DNA or RNA into the bacteria. To date, there are 14 different bacterial hosts. Today we are just looking at mycobacteria and microbacteria. Bacteriophages also have two different life cycles, which I will go into more detail here shortly. They have the lytic life cycle, which kills the host cell, and the temperate life cycle, which does not kill the host cell. Bacteriophages that have more than 50% of the same genome are in a subgroup called a cluster. And this is, when you hear me say clusters, these are, just think these are bacteriophages that have similar genomes. These clusters are designated by letters and numbers, so it could be like A1, A2, A3, or DD, DE, DF, that kind of thing. So, the bacterial hosts. These are what the bacteriophages use as a kind of a replication factory, if you will. And let me get my pen out. On the right is going to be Mycobacterium smegmatis, and you'll see like individual rods like here and here and maybe here. These are the different clumped together. So as I said before, this project is going to be based on Microbacterium and Mycobacterium. Some of these bacterial hosts will contain DNA primase and polymerases, which are required for the replication of both the bacteria and the bacteriophage. DNA primase will set primers, which are just short pieces of DNA, where the DNA polymerase will bind on the genome. So the DNA primase is responsible for the DNA polymerase binding, and the DNA polymerase is actually what's going to replicate the genome. It's going to read one strand and spit out the complement to that strand. So the repressor gene. This is what is going to determine the life cycle of the virus, as I was talking about previously. So if a bacteriophage does not have the repressor gene, it will be in the lytic life cycle, meaning it will use the host to replicate its DNA, and once it has replicated enough bacteriophages, it will lyse the cell membrane on the bacterial host and release the virus en masse, which essentially explodes the host, and all of these bacteriophages are released to go infect other bacteria in the surrounding area. So if you look at the figure, this is going to be your bacteriophage, and you see a ton of them coming out, and this right here is going to be where the cell membrane has been lysed. So that's where they're actually exiting out of. So if a bacteriophage does have the repressor gene, it will be in the temperate life cycle. Meaning it will also use the bacteria to replicate its DNA, but instead of lysing the cell, it will actually use it as a quote-unquote factory, where it will, as the bacteriophages are replicated, they will kind of sneak out of the bacterial membrane in order to infect more bacterial hosts. And doing this allows them to continue to use the same bacterial host to infect other bacteria. So in essence, the repressor gene just prevents the bacteriophage from lysing the bacterial host. So, Famrator is one of the tools that we used to be able to physically see the bacteriophage genome. Famrator has the genome of all sequenced bacteriophages on there if it's been uploaded. And if the genome has been what's known, you'll be able to see the gene. So these individual boxes of the genes, and you'll be able to see the gene names, which are the essentially just the functions of the genes. And I'll explain everything else. When you are calling a gene, you want to look for the highest E value possible and the least negative Z value possible. And then you're looking for the start and the stop sites. This start site right here for the terminase would be about 4,500, and your stop site would be about 6,400. So this block right here falls between about 4,500 and 6,400. That's your start and your stop site. Basing those start and stop sites off of genes that are similar to this terminase. So you would be looking for other genes that are similar to that. And then you're looking for the function of the gene. So based on the sequence of this terminase here, you would get a E value and the lowest E value, different E value than the first one. And the lowest E value you can find, the better, which it just means that you have a lower chance of that being caused by chance, that sequence being caused by chance. So you have all of these numbers above the genomes, right? Here and here. So if we look at the terminase again, which is right here, you have 17,675, which is going to be known as the FAM number. So the FAM number is a cluster of genes that are basically the same sequence. So you can think of them as the same gene in different bacteriophages. Some bacteriophages contain the same gene, and that's what that FAM is. It's just that family of genes. And then you see the number to the right, 73. It just means there's 73 sequences in that FAM. So another tool that we used was PhagesDB, which is also a free website you can use. So this gives you information about, so you'll see a picture of the phage right here. You'll see the host that it infects, the year it was found, where it was found, that kind of thing. So you can pull the whole genomic sequence of a single phage, or what we did was we pulled all of the sequences for the FAMs that we were interested in. So I have 10 FAMs that I was looking at, and I pulled the sequence of all of the FAMs, and then here shortly I'll show you a program called Eugene, and then we put all of those sequences into Eugene to look at them, to compare. So, what is the purpose of this project? We started by importing all of the repressor sequences that we found in the Mycobacterium and Microbacterium genome. And I'll show you how we imported that in just a second. But the purpose of what we were doing is we wanted to look at all of those sequences we imported and be able to rename and reorganize the nomenclature so it's easier to find the genes you're looking for. Because some of these, like I was looking at the repressor genes, and I had like 7 or 8 different names of the same gene, the same repressor gene, in different phages. Like one of them would be repressor, one of them would be repressor protein, one of them would be immunity repressor protein, or so on and so forth. So the same genes have different names, and we're looking at renaming these genes so they're easier to find all around. So the first step in starting our process was we had to learn a little bit of minor coding in the MySQL language, and we did this in the Linux terminal via a program called VirtualBox, which I'm not going to get into. So I wrote a command to pull all of the information I needed from the Famerator database via this command line. And the information I needed was the phage name and the phage cluster, the FAM that the repressor gene is in, the name of the gene, and by the name of the gene I just mean which number in the bacteriophage genome the gene is. So if it's number 34 in the genome sequence, that's the name of the gene, and what the gene was called as. So as I was explaining before, what the gene is called as is what's known as gene notes, and that's going to be things like immunity repressor, putative repressor, repressor protein, that kind of thing. So my command line obviously wasn't perfect because I still had to proofread the Excel document to get only the information that I wanted. So I essentially looked in the Excel document and filtered out all of the things that were not called as just a repressor or an immunity repressor or something like that. So these are going to be things like the CRO anti-repressor protein. If the gene note said anti-repressor or something like that, I would delete it out of the Excel sheet because that isn't the gene I was looking for. Some of the FAM numbers that were pulled from Famerator were also wrong. So as I was going through, I would take the FAM number that I got from the MySQL code, and I would put it in PhagesDB and then download all of the sequences in that FAM. Unfortunately, some of the MySQL and Famerator database FAM numbers are incorrect. So I would go into Famerator, and whatever phage I was looking at where the FAM number was wrong, I would go to that gene and see what FAM number it actually was, and then download all of that FAM. So as I stated in the last slide, some of these FAM numbers are wrong, and we have to go through and manually. So if we look right here, we see the FAM number up here in the top right. We see the start and stop sites here, and we see the gene name here and the phage name here. So the next step was to drag and drop the downloaded sequences into Eugene and then align them. This allowed us to look at how conserved the genome was. And by conserved, if you look at the picture below, this is the genomic sequence of amino acids for this gene. So this gene is made up of these amino acids in a sequence. So if you look at the, let's say this leucine here, this L, so it's dark blue. The color is dark blue, and that means that every sequence, or mostly every sequence that is being compared, has a leucine at that position. But if we look at this position here with the G, we see that every sequence except this, except the phage Sidious, has a G at that position, whereas Sidious has a D, and that's why it's white, because it is not a conserved amino acid in that position. So, looking at our first result, FAM6605, it had two sequences in it. The phages were found in cluster A4. The average sequence length was 55.5 amino acids, and the gene notes included immunity repressor, helix turn helix, DNA binding domain, and as you can see on the half of the genome from about here over, it's pretty well conserved. The left half is a little iffy. So for FAM 24303, so I'm going to explain this really quick. The number before the / is how many genomes I was able to pull using MySQL, and the number after is how many I downloaded from PhagesDB. So the 5 is how many sequences I personally looked at, whereas the 159 are all of the sequences in that entire FAM. So from the FAMs I was looking at, they were found in cluster A1, array setting, with an average sequence length of 131, and gene notes included immunity repressor and repressor, and they are highly conserved genomes, as you can see from the video on the right. So FAM number 24498, there were 3 sequences found in cluster A3, an average sequence length of 140, gene notes included immunity repressor, and was highly conserved in the first half. FAM 33982, the number of sequences I looked at personally was 1, but I downloaded 195. That one was found in cluster A1 with an average sequence length of 105. The gene notes included immunity repressor, and these genomes are a very spaced conservation. They're not necessarily more conserved in one half or the other, or one place more than the other. FAM 38858, I looked at 10 sequences with a total of 193 in the FAM. They were found in cluster A9 with an average sequence length of 144.1. Gene notes included a repressor and immunity repressor, and it is not a very conserved genome. So, FAM number 38075, I was looking at 1 sequence, but there were 48 in the FAM total. They were found in clusters A8. Average sequence length was 120 base or amino acids. Gene notes included immunity repressor, and it's a relatively conserved genome throughout the whole genome. FAM 4335, number of sequences I looked at was 4 with a total of 31, found in clusters A1, A4, and A11. There was an average sequence length of 123, but it was highly variable. Gene notes included immunity repressor, and the second half was well conserved. FAM 4338, I was looking at 4 sequences. There was a total of 8, and they were found in A6, A11, and A16. The average sequence length was 136, with the length highly variable. The gene notes included immunity repressor, and the first half was very well conserved. So, FAM 4388, the number of sequences I was looking at was 345, with a total of 413. They were found in clusters A1 through A7, A9 through A15, and A17 through A20. The average sequence length was 175.2. Gene notes included immunity repressor protein, putative immunity repressor, and repressor protein. This had a highly spaced conservation, with no one area in particular being well conserved. So, FAM 4566, the number of sequences I was looking at were 205, with a total of 260. They were found in clusters A1, A3, A8, and A18. They had an average sequence length of 176.2. Gene notes included immunity repressor, putative repressor, and repressor, and this was a highly conserved genome. In conclusion, the current system is disorganized because there's no specific nomenclature that is being used. People have tried to use a specific nomenclature, but it's kind of everywhere right now. This makes proteins hard to find, genes hard to find, and you don't really know what each of the functions of the genes are. This prevents experiments on those genes because they are not able to be found. It makes data collection hard. This work that this project is about can change that. It will make genes easier to find by giving a strict nomenclature for each gene, and it's not a hard process. This takes, maybe a week at the max to pull sequences and get all the sequences you need to rename, and it gives a lot of information at one time.